Today's lecture will be delivered by Mr. Sushil Vaidya, who is an application scientist for Forte Bio, Paul Life Sciences. He has worked in proteomics, lipidomics, small molecule characterization using mass spectrometry, HPLC and LC-MS based method development for biomolecules and small molecules. He has thorough understanding about the technical strategic planning in analysis of biosimilar characterization. In the next two lectures, Mr. Vaidya will have interaction with us about novel label-free biosensors. Especially he will talk to you about Bio-Layer Interferometry based platform through a lecture and a demonstration sessions. So let me welcome Mr. Vaidya for his lecture on BLI technology.
Sushilendra Vaidya. I am an application scientist for the Paul Forte Bio instruments. I take care of this applications parts in the entire India, where exactly the installation, the support, the training, as well as the product promotions. This is the alternate technology where exactly the Bio-Layer Interferometry, the interference based technology. The SPR is the one, the surface plasmon resonance, but this is the technology wise, it's a very different, and as well as the high throughput platform. So how this technology will be helpful in the interactomics kind of workflows, and I will go through what the technology behind the informations, how the principle behind this, how you can use this technology in your applications. So I will go through that. As you know that from the morning onwards, we are discussing why the biomolecular interactions are very important. If you look at in our body systems, most of the living organisms, whatever is happening in the systems, all through the transcription factors binding to the DNA, or the protein complex formations, or in terms of the signal transductions, where exactly the hormones and the growth factors and all what interactions happening, those things, and as well as the immune responses, the antigen antibody interactions. All this works on based on the interactions. And even when it comes to the drug discovery, where exactly we want to understand the mechanism of interactions, how the drugs is binding to the particular target. When we are discovering such kind of molecules, we have look at the affinity. So when it comes to the dose, I can give a simple example. Any drug, if you take it some drugs you have to take it three times a day. Some drugs you have to take it once in a week. Some drugs you have to take it in once a day. So how all these dose actually decides? So these doses depends upon the interaction platforms. If you say you have a target, when it is any molecule you have discovered, when it is bind, so how the affinity is, how strong is the affinity? If it is that affinity is strong, then you require a less amount of a drug. If it is you require a more dose, then you have to be the action will be it is clearing from your body very fast, then you require more doses. So how these will be helpful in your drug discovery areas. So in this, actually when you look at all the systems, interaction is the very important phenomena. So we need to understand, we need to characterize it. So if you look at what are all the conventional technologies people are in routine they are using, the ELISA based platforms where exactly people are using for the screening of the interactions. So when it comes to the limitation of this ELISA is like that, more it is a time consuming, and you require a manpower more on to develop the assays, and then you have to screen it further, and you have to select the right candidate for that. When it comes to this, it takes a long time, the limitation, and the reagent and the consumption of the reagents also more, and it is a more laborious. The other technology, if I look at, is the ITC, the isothermal calorimetry based titration, where exactly the interactions, when you can decide the stoichiometry of the interaction, as well as where exactly the parameters like delta S, where when any reactions happens in the interactions, where there may be heat released in the medium or heat absorbed in the medium. So those are the parameters we can find out. But what are the limitation with the ELISA and the ITC is like that. You can determine based on the concentration versus the response, then you can plot where you can read the steady state. But this will provide you the KD value, where what we quote as a KD is nothing but the affinity constant. But it will not provide you the kinetics parameters like on rates and the off rates. This is the limitations with the ELISA or the ITC based technologies. So why people are moving to the label free platforms is where you can get, this is the typical example when there is a ligand, say suppose the compound A and the protein A and the protein B, this forms a complex, and this is the forward direction. When the complex formation, finally it will dissociates to the once again A plus B. So these label-free interaction platforms are applicable to the reversible kind of reactions. So if you look at when any molecule binding, say suppose ligand A, it is binding to the analyte. When you see this is the response, this phase we call it as the association phase. When the same complex, when it dissociates back, so the complex will be dissociates, then you can see the dissociation. So the real-time interactions will provides the both on rates and the off rates. This is the kinetic information. So none of the any other techniques like ITC or the ELISA, it will give this information. So that's why this is a very important tool for the on rate and off rate determinations. So if I look at one of the example when it comes to the ELISA based on the steady state analysis, if you look at the KD parameters for this from the ELISA, it looks like both are same. When you performed on the label-free platform, if you look at the kinetic constant parameters, see this example, the blue trace, if you look at, it is dissociating very fast. When it comes to the pink one, the dissociation is very slow. Okay, so why this is important is like based on this, the rate of the dissociations, we can able to see the differentiation between when you are selecting the right candidate. The SPR technologies, the real-time label-free interaction systems, where exactly on the gold chip you have a matrix, to that you are coating one of the protein of interest, and then you pass the analyte through the fluidic systems, and then you can see the interaction change with respect to the angle. So when it comes to the limitation, when the SPR, when we are doing, you require like a dedicated operator for this. And apart from that, the time it required for the initialization and then get to the data, it is like a tedious. It's almost like a day it will take. So when you are doing such kind of a interaction, you require more patience, and to get the data, it's a more time taking and all. But apart from that, one of the important is the microfluidics delivery systems. When you are working with any kind of samples which are like a cell culture based, or if the sample from the body fluids and all that, you have so much cell debris or any other impurities and all. There may be a possibility of that they clog the system, the fluidic system. So that is very expensive in case of the SPR. If it is something get clogged, the flow cell is a very hairy like structure here typically. If it is clogged, then we have to replace the assembly of that. That is the very expensive, and the maintenance cost will be more. But when it comes to the other technologies, the BLI technology, there is a more advantage. I will discuss on that. And apart from that, the low throughput. Actually, here it is in the SPR techniques, you have a channel, you have to inject one concentration over the other. Serial dilutions you have to inject over the surface. But when it comes to the BLI technology, it's a very high throughput. That's why the limitations comes into that. When you are deciding the right candidate in case of the screening experiments, BLI is the more advantage in that. So when it comes to the BLI technology, I'm going to talk on this. This is the Fortibio is the parent company. They invented this technology in the year 2003 onwards, and the first system, if you look at the 2005, it has come into the market. They call it as Octet QK, and over a period of year, we have different instruments come into the market based on the throughput and all that. So with this, I can start what exactly the principle behind. If you take it, most of the interaction systems or any analytical techniques, all are based on the light. Light is except mass spectrometry. The rest of the analytical, if you take it analytical any, all are light based. As you know that light have a property, the wave property is the one. And the interference, when it works, the BLI comes, the Bio-Layer Interferometry. But the interference patterns makes the signal pattern here. As I mentioned, it works on the wave property. As you know that when the light passes through the fiber optic, there is some kind of a matrix. You have an obstacle. The wave, it is forward hitting that matrix and get reverse back. So what happens is, this is exactly the forward wave. This is the reverse wave. When it both forward and the reverse wave, when it is superimposed together, you can see there is an amplitude get increased. This is we call it as a constructive interference. When the same wave, this is the forward wave, this is the reverse wave. when it is, this is the forward and the reverse wave when it is opposite together at 180 degree. When what happens is that signal get cancelled here. So this is we call it as a destructive interference. So constructive interference and the destructive interference makes the signal pattern. So how we are doing that? So we are using a biosensor here, as in the SPR they use the chips. The similar way we are using the biosensor here. The biosensor, if you look at, this is the sensor, typically looks like a needle. This is made up of a plastic, and this is your glass capillary, which is nothing but the fiber optic. At the tip of this fiber optic, we are coating the biocompatible layer, it is the optic layer. To that optical layer, you are attaching one of the protein of your interest. So suppose you have two binding partners, one of the protein you have to attach on this. It is nothing but the solution-solid interface. Solution-solution interfaces are the, I can say that giving example, ITC, ELISA you can see as a solution-solid phase interface, because one of the thing you are putting on the plate. So these label-free platform also like a solution-solid interfaces. So one of the protein we are immobilizing on here, and then dip into the well containing a corresponding binding partner. So this sensor is a two-dimensional binding surface. The matrix what we have coated at the tip is the biocompatible. It is inert in nature. You can work with any kind of your physiological systems, buffers, or you can work from the pH ranges from 2 to 10, depending upon the application. So most of your biomolecular interactions happens at the physiological pH. And it is a uniform when we manufacture the sensor substances, it's uniform across the lots, and we test and then we releases. And whatever we have, the coated material is non-denaturing, it will not interfere with your interaction system. So if you took the diameter of this, the optic fiber is just only 600 micrometer. You require a very less amount of your sample to immobilize on the sensor surface. So if you look at how this exactly the instrumentation inside on the Bio-Layer Interferometry, the octet platforms, this is the spectrophotometer here, and it is connected with the robotic arm, and these are the sensor tray. I can, if you look at, this is a 96 well plate form. So the robotic arm picks up the sensor and dip into your 96 well plate format. So the sample plate, we have an orbital shaker and the temperature control. What is the difference between the SPR and the BLIs? We don't have any microfluidics here. Just it works on the dip and read. It's pick up the sensor and dip into the well. So everything happens, the reaction, whatever is happening at the tip of the sensor. So in case of the microfluidic devices like SPR, the flow assists in the binding. But here we have an orbital shaker which assists in the binding. So when it comes to the high throughput, we have different channels. Like in SPR, if you take it, there is a 4-channel, 2-channel instruments. So where in exactly the 4-channel means you can pass the 3 analytes and 1 acts as a reference. And here we have an instrument, we have an 8-channel here. 8 interactions you can measure simultaneously. That's where the throughput comes. We have a 96-channel here. 96 interactions you can perform in one goal. And we have a 16-channel, 16 interactions you can perform in one goal. We have a single channel also, 2-channel, even we have 2-channel automators instrument. So how exactly, what the principle behind this? As I mentioned, it works on the interference based.
Sorry, going back to that previous slide. So is the right hand the on rate and the left hand the off rate, or how are you doing binding and then how are you looking at dissociation?
Okay, so the sensor, this is the sensor compartment. The sensor actually pick up the robotic arm, pick up the sensor and dip into the well containing the first buffer. And I will show you in the subsequent set how it, okay. So what exactly the principle behind this? As I mentioned, it's interference based. We are passing a light, it's just a white light. Light get reflected back. If you look at, you can see the reflections coming from the one internal layer and one from the ligand end. Just I take it, I had immobilized one of the protein of interest on the sensor surface, then I dip into the well containing buffer. If you look at all this, what exactly? It is amplitudes, different amplitudes we have, constructive interference, destructive interference. This is what we plot, relative intensity versus the wavelength. So what happens? So if the one which have a higher amplitude, where exactly the waves get superimposed, then you can see the amplitude get increased. Where exactly there is a partially superimposed, you can see this kind of, and there is a destructive interference, there is a completely signal get cancelled. So what happens when the same sensor, when you dip into the corresponding binding partner, now you can see that earlier was just only light reflecting from it's a blue colored layer. Now you can see there is one more orange layer. If you see this, now right, which is reflecting from the orange layer. Once again, if you look at all these waves are there. Earlier was the plot like this. Due to the molecule starts binding to the sensor surface, you can see there is a shift in the interference pattern. This is what exactly, as the molecule binds to the sensor surface, you can see the shift. So what happens exactly on the tip? The molecules bind to the sensor surface, it forms a biolayer. It depends upon the thickness of the biolayer. More the molecules bind to the sensor surface, more the molecules bind to the sensor surface, you can see the relatively shift in your spectrum towards your red. This is what exactly the principle. So using this phenomenon, as the more molecule you can bind, you can see the correspondingly it relatively shifts towards the right. So you can do the quantitation using this phenomenon. Not only the kinetics, you can determine the quantitation. So in real, what happens? Molecules bind to the sensor surface. You can see the real-time picture. As the molecules binds to the, then you can it leads to the equilibrium. Then the same sensor, when you wash off the bound molecule, you can see the dissociation. This is what exactly the real time you can look at. The background is this one. The molecules bind to sensor surface, then make a biolayer and relatively shifts, and once you washed off, then it will come back.
The Octet system from Forte Bio provides a complete label-free solution for analyzing protein kinetics and quantitation. The disposable biosensors measure binding events directly in a standard 96-well microplate. A proprietary protein coating at the tip provides reproducible coupling of target molecules and a minimum of nonspecific binding. Here, we are using streptavidin-coated sensors to set up a 5-step kinetic analysis where the first protein of the pair is biotinylated and we measure the binding and dissociation rates of a second molecule. With the sensors arrayed in a standard SBS format, some steps can be performed outside the instrument, like conditioning the sensors before the analysis. Up to 8 sensors can be run in parallel, and the menu-driven software assigns identities to each sensor to be used. In our experiment, each column of the sample plate is loaded with reagent so that we can analyze 8 samples in parallel. Firstly, a buffer to establish the baseline, then a loading step with the biotinylated protein and a second buffer for a new baseline. The binding protein is being presented in a crude cellular lysate, and the dissociation step will be measured in buffer. Once the sample plate is loaded, the experiment is defined in software so that an appropriate analysis will be made at each step. Sample identities and whole method files can be stored and imported to simplify or standardize routine analysis procedures. Data files and results will be stored in a predefined location to be analyzed at the instrument or at a remote terminal. The sample plate is heated to the selected temperature, and a flow across the sensor surface is established to overcome mass transport effects. Using optical fibers, Octet feeds broad-spectrum white light down the sensor and collects the reflections. One reflection comes from an internal optical layer and one from the interface between the streptavidin layer and the surrounding solution. Most of the light scatters in the surrounding matrix. In a process that we call Bio-Layer Interferometry, the 2 reflections generate a spectral pattern that is collected and analyzed at the spectrometer. Constructive interference gives intensity peaks, and destructive interference causes intensity troughs right across the visible spectrum. As a calibration, the position of the interference pattern is measured for all 8 sensors, and then shifts in the pattern are plotted in real time. The streptavidin sensors are loaded by transferring into the wells with biotinylated protein. Bio-Layer Interferometry is insensitive to changes in refractive index or pH of the matrix. When more molecules bind to the streptavidin layer, the optical thickness of the layer changes. And as the thickness changes, the interference pattern shifts. 1 nanometer of protein binding to the sensor produces a 1 nanometer shift to the blue in the interference pattern. and a 1 nanometer change in optical thickness is recorded for that sensor in real time. The change in thickness relative to the initial calibration is plotted for all 8 sensors. With real-time monitoring and 8 samples run in parallel, it's easy to optimize protein loading so that it can be performed outside of the instrument. Once the biosensors are loaded, they are transferred to a buffer solution to establish a new baseline. There is no dissociation observed from the tightly bound biotinylated protein. Bio-Layer Interferometry is relatively insensitive to matrix effects, and Octet has no microfluidic constraints, so measurements can be made in very crude samples. Culture media with serum, periplasmic extracts, or cell lysates with particulates can all be measured easily and accurately with minimal interference from the matrix. These crude protein preparations exhibit different binding rates, and the real-time data output allows you to monitor the behavior of the proteins as well as the overall progress of the experiment. The process is so simple that it provides a convenient tool for protein quantitation in complex mixtures. The dissociation step may be a 5-minute off rate for a quick screening experiment, or 2 hours or more of data collection to determine a KD for tightly binding pairs. Octet is simple enough to use as a screening tool, and 5-minute off rates can be measured for 96 samples in little over an hour, providing ranking data for clone selection. Alternatively, a tightly binding pair can be monitored for over 2 hours, limited only by the evaporation of the sample. After the dissociation, the biosensors can be ejected and new sensors selected without any need to run regeneration methods or run cleaning protocols. All of the sample solutions are recoverable, or they can be reused in the same format with new targets depending on your experimental protocol. Because Octet is so easy to use, assay development is rapid, and you can quickly generate the data you need to make clonal selections or optimize a purification process. For accurate kinetic determination, single-use sensors provide precise answers without the need to work up regeneration protocols. For higher throughput screening and yes-no determinations, reuse protocols provide a cost-effective solution. And once you have developed an experimental protocol, you can automate the whole process all the way through to data analysis. At the end of the experiment, select Data Analysis and you have access to a complete suite of software tools. Curve-fitting algorithms from Origin allow for the reporting of binding and dissociation rates together with display of residuals and data tables. Or you can export results into your favorite program for further analysis. With a range of biosensor surfaces, including streptavidin, protein A, and amine reactive sensors for custom protein coupling, Octet provides a complete solution for protein kinetics and quantitation.
So what exactly information you can get from this? So as I mentioned, the one based on the shift you can determine the concentrations. You can do the direct one-step kind of binding, sandwich. If you want to convert ELISA platform onto the instrumentation, you can convert it. So depending upon, you can determine the micromolar concentration. You can go as low as the nanogram or the mg per ml to picogram per ml. So the quantitation you can determine, and the kinetics. The kinetics, as I mentioned, on rates, off rate, and the affinity constant you can determine, irrespective of whatever the biomolecules. It's maybe a protein to protein, protein to small molecules, protein DNA, protein RNA, and all that. Apart from the specificities, where exactly you can do the functional testings, functional testing with respect to Fc gamma interactions, where the people are doing in the drug discovery, the rank ordering when you're doing the hybridoma screenings with the large map platforms, where you can provide which is the best antibody will bind to the target. Based on, you can give the rank ordering on the off rate of the molecules. And the epitope binning, we have a dedicated software for the epitope binning. You can do the screening of your where exactly your molecules binds to that, and the isotoping also, subtyping of your the IgGs. So we have a lot of applications on this. I will go through later those applications. So one of the important parameter is most of the people, you people are doing the quantitations based on the Bradford assays or the total protein content. When you say, suppose you have a protein in a kind of a matrix where exactly you are expressing protein of interest in a cell culture, or you want to determine in the patient samples for how much my protein of interest which is there or not. So you can easily, because it works on the dip and read. There is no such kind of say, even though if it is cell debris are there, impurities are there, anything is there, it is purely an affinity based interactions you can easily quantitate using this phenomenon. So that's why the advantage is like that you can go with any kind of sample matrix. It is need not like that you have to go for the purified one. When it comes to the SPR, you have to go with the more purified samples. And the second advantage is like that you can go with the pH ranges easily like 2 to 10. Depending upon the application, you can screen quickly because it's have a high throughput 8 channel. You can quickly screen the samples, which pH is the favor for the binding. So these are the platforms we have. These are the 16 channel instrument, this is the 2 channel instrument, this is the 8 channel instrument, and this is the 96 channel instrument. Depending upon the high throughput, what required, you can choose the instrument in that. And the more advantages like that, as I mentioned, there is no clogging, it works on the dip and read. And the high throughput, you can save your time, you can quickly screen the experiments. And the good thing is like that when it comes to the interactions where exactly your samples have a DMSO or the glycerol. When it comes to SPR, presence of the DMSO and the glycerol, sometimes you can see the bulk effects because SPR is very sensitive to those changes. But with a dip and read, you can easily go with such kind of there is no interference from the glycerol or the DMSO in that. And the good advantage is like that it's typically the software is so user friendly. Just if I train for half an hour, you can start your experiments. No dedicated operator required for this. Typically, the programming is like your ELISA plate reader. So what are all the ranges, what kind of molecules we can go? Except cells and atoms, we can go in the blue areas. All these biomolecules, we can go with the interactions. I can show you the cells. Recently, we have got the publication from the GenMap. I'll discuss with those things. So these are the different applications we have: segregated, kinetic applications, quantitative, screenings, and development, assay developments, and all that. We have publications wise, we have more than 1,500 from the past 10 years we crossed, all published in a different impact factor journals. So how exactly it works? If you look at the animation, if I say this is typically a protein quantitation, if I take it any affinity based, so suppose you are working with one of the histag protein, how much is expressed in your culture. So suppose if I have a standard in that, if I dilute in the plate, 96 well plates, lower to higher dilutions, take 8 sensors, when I dip in this, you can get a curves like this. Based on the binding rate versus the concentrations, the higher concentration have bigger and the lower ones are this. When the same sensor, if I go for the regeneration, so where the bound analyte get washed off here, and then when it dip into the unknown solutions where your cells means your protein is expressed, you can easily quantitate using this plot. So very quick experiment in just in 15 minutes you can determine the concentration of your protein of interest. So how exactly works the kinetics workflows? First, the sensor will dip into the well containing a buffer. You get a baseline here, and next it will comes to dip into the well which have the loading, and then goes back to the well containing a buffer. There exactly the unbound material get washed off, and then corresponding analyte it is binding. You can see the signal and goes back to dissociation. The very simple. It's just you have to the robotic arm back and forth it will move. Just I will explain once. The buffer, the sensor dip into the well containing a first line of buffer, you can get just a baseline. There is no any molecule binding to that. That's why you get a baseline. When the same sensor, when it move to the next well containing a capture molecule, one of the protein of interest you have to immobilize on the sensor surface, you are putting in the second well, then the sensor dip into that well, you can see the loading response. As I mentioned, some molecules bind to sensor surface, you can see the change, right? The phase shift. So you can see the loading response. When the same sensor move back to the well containing a buffer, here just a baseline, there is no molecules binding, unbound molecules get washed off here, and the sensor next move to well containing a buffer, the analyte, you can see the response. And the same sensor goes back to the well containing a buffer, bound analyte get washed off. Very simple to operate, this experiment, you can quickly screen just in a 15 to 20 minutes, you can get the kinetics data. So we have a, on the shelf, we have a different biosensor chemistries with us. Depending upon your protein of interest, you have a tag, or you can choose, or if you don't have any tag, you can go with some kind of modifications like biotennylations or the amine coupling you can immobilize. So these are the pre-coated for the antibody platforms. We have protein A, protein G, protein L, and some FC capture sensors. And for the His tag based proteins, we have a nickel NTA, we have anti-His sensors. You have a GST tag basis, we have anti-GST sensors. And if there is no any tag your expressions, then you can biotennylate your protein of interest, and then you can couple to the streptavidin sensor, the biotennylated protein. We have a APA sensors, aminopropylsilane, for the hydrophobic interaction. Your protein is more hydrophobic, no need any tag or anything, you can just bind to the protein of interest on that. And for the superstreptavidin, we have this is the quartz based sensor, especially for the small molecule protein interactions. So we come to the application side, how we are going to use. One of the example here is, this is one of the group they are interested in developing the vaccine based on the influenza. They identified the 2 antibodies which are broadly neutralizing capacity to the influenza. In this experiments, what the author did is he isolated a 15 group 1 and the 44 group different hemagglutinin he had taken from these groups, and then he did a biotennylation, and then he mobilized using the streptavidin sensors all the 59 hemagglutinin to that, and then screened with the respective the antibodies. Among that, if you look at these, you can see the binding interactions of the different antibodies to the respective hemagglutinins. From this, they had developed the broadly neutralizing of the 2 antibodies in that. So very quickly, we can screen this kind of applications using this. So next example I can show is where we can understand the protein structure and the function. Here, what is the LET7 is the one of the important tumor suppression microRNA. So in this, what author did is the KSRP is the protein which is binding to the LET7 precursor. So what happens is here, the KSRP is, and the LET7 is your microRNA which is biotennylated and immobilized onto the streptavidin sensor, and this KSRP have 4 domains. So among that, he did a mutation in one of the protein, the domain KH3. If he did a mutation, it is not binding. So with a wild type, he had showed that it's the KSRP is binding. In the 4 domains, among the one KH3, if he did a mutation, it is not showing a binding. So using this, he concluded that with respect to this, KH3 domain is one of the important parameter for the binding of the KSRP protein to the LET7. So it is one of the important the transcriptional factors for the cell differentiation, and very important in case of the recruiting the oncogenes for the where exactly the cell differentiation happens. If it is a mutation happens in that particular gene, there may be a chance of the carcinoma and all. So one more where exactly facilitated binding interaction studies here. So this is one of the example where exactly the fuse system is the one which is important in the cell differentiation. Sysmic is the important regulatory gene. So this SIC means the expression of this particular gene is more in case of the cell differentiation. So how this happens, this regulation? So the author concluded using this, there is called as a FBP protein, presence of this FIR only interacting to the FBP protein. So in this experiment, what he had done, the biotennylated the fuse 40 DNA, he immobilized onto the streptavidin sensor, and then he performed with the binding of the FIR. So in presence of the FBP protein only the FIR is binding. Without this proteins, it's not binding. So he concluded like that. So very important tool when you do the in case of the basic research, you can quickly screen those. So one more example I am showing here is the folding and unfolding patterns of the proteins. So in this example, the sensors where exactly the biotinylated, the GROEL is immobilized on the sensor surface. The unfolded protein will be bind very then you can see the response. The folded protein will not bind. So using the chaperone model, you can use for the screening whether the folded or the unfolded patterns you can easily study. And the one more example, in case of the diagnostic immunoassays, giving an example here is like that the kinetics parameter 15 anti-HRP2 antibodies they measured. They compared with the ELISA where they had comparison they had done. You can quickly screen which of the pairing, the antibody pairing, which have better diagnostic for the diagnostic applications. When you are developing any ELISA kits or you require a primary antibody and the secondary antibody, you can quickly screen using that. So which are the best based on the kinetics profiles. So aptamer screenings. So aptamer is one of the area now is picking up comparison to your antibodies. Most of the many laboratories in India, they are working on the aptamers. So aptamers also similar, they have a different size and the shape. When it binds to the particular the target, you can see the response. So aptamers also have a major application in diagnostic industries. So one of the examples, you can quickly screen using the aptamer interaction studies using the BLI technology. So the one more important where exactly when it comes to drug discovery applications, small molecule protein interactions, if you look at with a one concentration different, you can screen as many as compounds as possible, and then you can look based on the off rate, you can select the right candidates. This is the one of the work from the University of Michigan. They had done a publication on this. They have developed the 3 hits with the complete kinetics characterization using this the bromodomain 1 protein. And the one more example I can show is the characterization of this AMPK protein to the 2 compounds, thalidoxine and the one more here. So thalidoxine and the structure isomer and the hemandazine, it's like compared to this, the thalidoxine is the more binding compared to the other platforms. You can quickly screen using this tool. And one more, the next very important when it comes to the ligand fishing experiments, I think it is more relevant when it comes to the interactomic studies. Because what is exactly the ligand fishing is? So suppose you don't know what is the protein is interacting to your one of the protein of interest. So suppose you take it, the protein of interest, when you can immobilize on the sensor surface and then put it into the cell extracts or lysates or anything, which is the protein is binding to the sensor surface, you can elute it and then you can go for the mass spec studies. So one of the example I am showing is the interaction, you dip it and then you can elute the binding combinations. And you can go for the electron microscope or you can go for the mass spec studies. So here one of the example I am showing is Montinza anthracotoxin prepore. This example, they activated the tip and they had immobilized the protein of interest, the anthrax prepore, and then they had processed with 1 molar urea at 37 degree. The prepore to pore conversion happens, and when they dip into the lipid colate mixtures, you can see the micelle formation between the pore and the lipid. So what they had done? Removing the biosensor tip, and they dip in a buffer and decoupling reagent, then preparing for the EMI grid. You can look at the electron microscope how the complexes are those. So you can easily perform such kind of experiments, because when it comes to the SPR, when you elute the complex mixture and when it comes to when you are going to collect that, because your sample is more diluted, then you have to concentrate it, then you have to perform the further studies. You can enrich in the dip in the well itself here. So one more example is it is an industrial work. The erythropoietin, they were expressing the Novo Nordisk company, where exactly happens when they are doing the quantitation, they are getting the concentration very less. Then they thought that what happening in that. So they identified some host cell histones blocking the EPO binding. So, they eluted those mixtures and they perform the mass spec with that. Then they identify that the protein of interest is binding to the, it's blocking the binding of the EPO to that. The GenMab recently has published this data. They immobilize this cell, the entire cell on the sensor surface. And they had tried the different combinations here. One of the example I can show you is they had taken a collagen here. They immobilized the collagen on the amine coupling sensor, and then the cell which is the A431 cells, which is expresses the EGFR and the beta adrenoreceptors, overexpressed receptors. They put it the cell on the sensor surface and then perform the interaction studies with respect to the EGF here. You can see this, the binding interaction. It is not exactly the kinetics. It is orthogonal to your cell based assays. So, what happens is, say suppose it is such a cell, it is a big cell you are immobilizing on the sensor surface, then a tiny like some protein is binding to a sensor surface, you can't see a biolayer change on the sensor surface. But what happens is, when any molecules bind to the respective target on the cell surface, you can see in the cell, you can see there is some events happens. There is a actin modulations or any other pathways. So, these changes will leads to the signal change on the sensor surface. We call it as a dynamic remodulation, the word. So, using this phenomenon, they had done the quantitation kind of stuff, the cell based assays orthogonal to that. And very important when it comes to the ELISA and the HPLC comparisons, where most the industry people, map platforms people are working with, they are using this platform for the titer determinations for their antibody productions. Very quickly, the good advantage is like that 96 samples you can screen just in a 40 minutes, or I can say the quantitate, the 40 minutes. But in comparison to your protein A HPLC or the ELISA based methodologies, it will take a long time. ELISA it roughly takes around 3 to 4 hours, but even protein A HPLC for the 96 samples, it's like 8 to 10 hours it will take. But you can just in a 40 minutes, you can quickly titrate the protein of interest in this. So, one of the comparison here they had done the early clone selection cell line developments. They had done a correlation with respect to the HPLC versus the octet platform. It's comparable with the other methodologies. So, depending upon the applications, you can choose the right sensor. You can put into different all applications. Either it may be protein DNA, or protein RNA, aptamers, or cell based assays, or the nanoparticles. We have a lot of applications in that.
So, I hope you got a better understanding about this label free biosensor, Bio-Layer Interferometry technology BLI. The next lecture will include a demonstration session that was conducted during this workshop. I am sure you will be now able to understand these concepts much better, and you will also understand how to perform these experiments for your biological samples of interest, for your biological problems of interest. Thank you.